What's up, guys? This is Jake Norley. He's been at Scioto Biosciences, and then now he's at Beckman Coulter, and he knows more about the gut microbiome than anybody else that I know. And like for him to get started, let him tell us, enlighten us.
Yeah, for sure. So, like Bob said, I was at a microbiome company, Scioto Biosciences, doing startup work for a novel activated bacterial therapeutic. Essentially, we just kind of modified the bacteria a little bit to basically be more efficacious by the time it hit the gut. Sorry, one sec. So basically, when bacteria, as they exist on the market today, they're lyophilized or completely dried down. And so that is a stressor on the bacteria. And while they're still alive and they're still efficacious in work, when you're first taking that bacteria, it actually is rehydrated in your acidic gut. And so it's not necessarily known how many of those bacteria actually pass through from the stomach into the gut and the inner lining. So the whole foundation of our product was that we were giving a activated bacteria that had already been rehydrated and was grown a little bit so that it could actually weather the hard conditions of the stomach and then get incorporated into the intestinal lining. And
the kind of modifications you're talking about, it's more like you're putting them into a material, or it's not like a genetic modification,
right? Correct. So it's not genes or DNA altering. We just took a strain that was pretty commonly used, and we allowed it to incubate for a certain amount of time with particular materials like Cefidex and some prebiotics, and really got a mucus-like environment for the bacteria to actually start expressing the proteins that they would be expressing when they're actually in your gut, or when they would adhere to the gut lining. So that's kind of our product, and they're actually still working. When I was working there, we had our IND approved for phase 1 clinical trials, and we kind of found some really interesting data, to be honest. And there's a lot of data coming out on this. But it's the brain gut connection, and so there's a lot of data that 5 years ago even a lot of scientists would have just said was hearsay, said is doesn't make sense, like we don't understand how that works, so I don't believe your data. But we're actually seeing some really rigorous studies that have been done that show specifically with the bacteria we're working with, Lactobacillus reuteri, it will increase oxytocin levels in the blood. So when administered, we read a paper that had done something very similar, and we realized we were working with the same bacteria, so it made sense to start testing the same things. And we saw that we actually did have an increase in the oxytocin, even compared to the control regular lyophilized bacteria. We saw a bump up in our activated bacteria. So we know that in some fashion, these bugs are creating some precursors, or maybe even the scaffolding for that signaling hormone, and that is somehow translating through the epithelial cells and into the blood to actually have some profound effects on the neurological systems. So we pivoted, decided to target autism because oxytocin is known as the love hormone or the social hormone, and can actually be really vital in a developing brain. And so we thought that with this sort of very safe GRAS approved sort of method, we could try to uncover a little bit more. And as we were going through the science, as typically happens, funding kind of fell through, and experiments went wrong, and so on and so forth. So for the company to continue on, they actually had to let me go, which is why I'm at Beckman Coulter now. But the super interesting thing I learned in my year and a half there at Scioto is all of the neurological connections that actually come from your intestines specifically. And it's a little cliche, but you are what you eat is kind of very true to everything that you do. Taking one medication, one little pill once a month or once a week or once a day even, kind of pales in comparison when you look at the amount of molecules you're putting into your actual system via your food. So that has to have some sort of, effect in all of the things that you've been going through with how complex it is with insulin signaling and everything else.
Yeah.
Obviously these little critters in our system are having some sort of profound effect in everything that is, a human, so to speak. So that was a super interesting find, but I'll talk a little bit more specifically about the gut and like what I've learned from my experiences there at Scioto. It appears that, from my understanding, when I came into Purdue, I had some conversations with people about the brain-gut connection a little bit, and it seemed that most researchers just kind of view it as a black box. They're like, this is a box of uniform, just chaos. There's a bunch of different bugs, and they're all in there, and we don't really know how to study it because there's trillions and trillions and trillions of different species and genus of bacteria. So how do you even, begin? And so we were kind of walking around in the dark. There wasn't really labs that were doing what we were doing or really knew how to study the gut. And there's, still people out there that are figuring it out. But basically we, read up everything that we could and figured out that, it isn't just a black box. There's a lot of structure to it, and it's all harmonious and works together. So the bacteria that we were working with is called Lactobacillus reuteri. So the Lactobacillus genus is typically the first bacteria in your gut lining. And so it is not completely anaerobic. It thrives a little bit better in non-oxygenated environments, but it can be that buffer there because it can exist in environments with oxygen. And so at the front lining of the gut, these Lactobacillus actually will form together exopolysaccharides, or exopolysaccharide chains. So it'll take all these different sugars and just start connecting them together. And so this forms an extracellular matrix around the bacteria. And so the bacteria will continue to do this and just, build up these chains of energy and sugar. And it helps to protect the bacteria. It helps to coat the lining of the gut and the epithelium to make sure that no pathogens or anything are getting through. And then on top of that, because it's kind of the first bacteria in the gut, we kind of hypothesized that these exopolysaccharide chains that it's making is kind of going further down the line and then being used as energy by the bifidobacteria and then subsequent bacteria to continue to thrive and keep going, doing its thing. And so bifidobacteria is also another good bacteria or probiotic that you've most likely heard of. If you go to the store, you'll see when you flip over the back of your probiotic, there'll be a bunch of CFUs of lactobacillus this, lactobacillus that, bifido. So those are the good bugs. And there's obviously some bad bugs, but really these bugs kind of just work in harmony with our bodies, and we've kind of evolved from the ground with them. And without those bugs, we would not be here because, as I alluded to earlier, there's obviously more complex interactions that are occurring here when we're seeing that hormones that are vital for our neurological development and vital for us really understand or really just kind of growing and moving forward with our brains and everything are being influenced by these bacteria specifically, not even a change in the diet or what have you. So there's really a lot to unpack here, and I think we're just on the forefront and the cusp of what is this brain-gut connection, what is the microbiome, and how is it going to affect us and our health in general.
So if I'm understanding correctly with L. reuteri, if you didn't have enough L. reuteri, then there's nothing to feed the other bacteria because they're the ones that produce the exopolysaccharides.
So there's a lot of different lactobacillus, and they all kind of have this morphology of making an exopolysaccharide chain. L. reuteri we kind of focused in on because we were really interested in necrotizing enterocolitis, which is a disease for underweight babies, where basically they haven't gotten to full mature term and they don't have the correct bacteria in their gut. And basically what happens is the bad bacteria take over and start necrotizing or eating at the intestines and everything in there. And it's a 50% mortality rate. And the current surgery is you have to go in and cut out the dead intestines and then stretch the intestines the rest of the long way, and hope the baby survives. So there's a real importance to having a correct microbiome. And L. reuteri is actually found in mother's milk. And it's one of the real foundational cornerstones of, starting out life and starting out a good sort of gut health. And a lot of the data we found really supported this. And it was really interesting to think about it chronologically and think about, the GI tract in general as like, what needs to happen in order for a healthy gut. And this, first part of it was basically where we started, because we're looking specifically at the beginning of life, looking at specifically helping these babies to, not even begin the necrotizing process. And hopefully, the good bugs actually create that environment to allow the good bugs to thrive over the bad bugs.
That makes sense. So like when the good bugs are thriving as opposed to the bad bugs, is there something that the good bugs do that kills the bad bugs, or like how does that work?
There are, so Lactobacillus reuteri actually does produce not an antibiotic, but an antibiotic-like compound specifically for the bad bugs. I think it's Reuteri, which is kind of why the strain was named Lactobacillus reuteri, or maybe vice versa, I'm not sure. But Reuterin's that molecule that kills some of those bad bugs. I think the bigger part of it is, though, that these good bugs just more so take up space. So we found that we did some other studies about C. diff infection, and so we think that it could be efficacious for C. diff as well. And so C. diff is another bacteria that's just a bad bug, and it comes up in hospitals a lot, especially after an antibiotic regimen or something like that, where you're wiping out all of the bugs there. But C. diff is particularly hard to kill. And so my theory is just kind of that if there aren't the Bifido, Lactobacillus, and other bugs to occupy space in the intestines, then these bugs have the opportunity to start to latch onto the epithelial cells themselves, because there's less of the bugs to produce that mucous membrane and to just take up space in that epithelium.
That makes sense. So like they kind of produce like the mucus in the little areas by taking up all the other space. They produce the little areas where the other bugs will grow. But if they're not around, then those other bugs will go after the epithelial cells, and then it just goes south.
Yeah.
That makes sense. I actually wanted to like also go towards like obesity. Do you know anything about like how the gut microbiome might affect obesity?
Yes. So interestingly enough, I was actually on a call earlier today, and basically I was looking at this graph, and it's talking about mortality rate in respects to COVID. And so, I had a huge spike at the Spanish flu, and mortality after that pretty much decreased all the way until about like the 1970s, mid 1970s or so. And then you saw a little bit of an uptick. Nothing as drastic as the virus is inducing, but given our advancements in technology and everything else, you wouldn't really expect life expectancy to drop off, but it kind of has. So basically with obesity, there's a lot, like I do have a lot of kind of hypotheses on this. The glucose reward system is kind of our most primal dopamine pathway. And also it's very socially acceptable to, eat food, and you obviously want everyone else to enjoy good food.
Yeah.
But with the excess of added sugars with processed foods and things that actually have been shown in studies to destroy some of your gut microbiome, some of these, foods that you're taking in could, or most likely do correlate with obesity, right? And you kind of have this addiction pathway going, and you get people that are just so fixated on food that they end up over consuming and becoming obese. But I do know that a lot of these processed foods and added sugars and things of that nature that are not what we were intended to consume in mass amounts. It's here and there and fruit and other things that weren't necessarily that plentiful, but now we've kind of hacked the system and figured out how to just add that to everything. And so, since those 1970s or so, we've just seen an increase of diabetes and cardiac disease or cardiovascular disease. So I have to imagine that there's some sort of correlation here. And I recently read a book called Food Fix by Dr. Mark Hyman, Hyman. Ironically enough, my boss at Scioto's name is also Mark Hyman, but Hieman. But anyways, this book really just dives into all the causes of or basically just the food industry in general. And it is a real big advocate against, added sugars and processed foods, and more so organic and, homegrown kind of foods on many different levels. It goes into agriculture, politics, and the like. But obviously my focus and interest was the science of what's happening into your gut there, and how are these added sugars affecting you? How are these, processed things affecting you? How is glyphosate affecting your gut microbiome? It's a thing. If you don't know, glyphosate is a pesticide that's put on pretty much everything, and regardless of where you live and where your food source is, it's pretty hard to not consume. There's a lot of glyphosate in everything that we touch, essentially, and it actually has very detrimental effects. So these small molecules that we're ingesting in mass have to have some sort of effect on the bugs that live within us, and that are supposed to be helping to aid our nutrition and help to buffer that lining in the gut to keep inflammation down. And basically that's kind of my thoughts on it. And then once you start to kill off those good bugs, right, the lactobacillus, the bifido, and there begins to be more space that's just unoccupied in your epithelium, there's more entry points for pathogens to get in. So if you're eating things that are causing your good bugs that are producing that mucous membrane and that protection barrier there, you're eating things that are killing those bugs off, then there's just more access to the body in general. There's going to be a heightened immune response. You're going to have a lot more inflammation, and your body's going to be less prone to behave in the way that it should, to cognitively function at a high level, to fight off infections, heal cuts and wounds, so on and so forth.
That makes sense. So I remember earlier you were talking about the gut-brain axis, and we also you also talked about just now about the glucose pathway and how that releases dopamine. Do you think the gut microbiome could actually affect that, and is that the reason why somebody might become obese as a result of it?
I mean, quite potentially. I think that the obesity problem is it definitely is an addiction. It is something where you find solace in food and you go to that and you overindulge. But also it's an issue of the food industry as well, to pack as many, good tasting calories into one little thing. And then you end up with people who really can't afford to eat good food, eating the cheaper, delicious food that has been manufactured to be delicious. And then you kind of, see that rise in obesity a little bit. I had another point on that, the addiction line there. It's escaping me now, but that reward pathway is just so primal in us, and it's something that you always go back to. What I was going to say is, in society, you look at a heroin user and obviously you condemn them. You condemn their activities. You say, oh my gosh, how'd you get to this point? I can't even fathom it. Even with alcohol, it's kind of the opposite. It's oh yeah, we all drink, we all have fun, this is acceptable. And so you have a lot more alcoholics than you might have heroin users, but you can still function at some level of alcoholism in certain cases. And then when you come to a topic that is not even looked down upon really at all, just breaking bread and eating food together. When your food has changed so much from what it was that your grandparents ate, and it's a bunch of this manufactured to taste good food that has a bunch of processed chemicals, a bunch of added sugar, and things like that, that's when, I think that addiction really takes hold when your society says, hey, like these are things that are okay to deal with your pain, because no one's really condemning anybody for eating. That's when you kind of have the obesity epidemic that we have, where you have like validation to your addiction that allows you to continue without any sort of repercussion.
That makes sense. All right, so going into it further, I did a video about the gut microbiome where I looked at 3 different papers, and it was kind of interesting that like antibiotics previously, like in animals, they would use it in farm animals to make them bigger, and as a result, you get more meat from the same animal, and they were optimizing for that. Do you think
myosin inhibitors?
Say that again. Sorry, it just like bugged out a little bit.
So yeah, you froze up a little bit. Was that a myosin inhibitor drug that beefs up the muscle, or is that a different?
No, they were just using like in the past, like throughout the 1950s up to around the 2000s, they would feed antibiotics like bacitracin. They would feed antibiotics to the animals, and the animals would just get bigger. So is there something like, the way I was thinking about that is the antibiotic, it's a broad spectrum antibiotic, so it would go in and it would just like kill all the bacteria in their gut, and as a result they would get bigger. Is there a reason to think like not having bacteria in your gut would actually force your body to start storing instead of like burning what you eat?
It's interesting. Yeah, I haven't seen that paper specifically on antibiotics, but I would imagine that if you're killing off all those bacteria in your gut, there's trillions of them, and they're helping you break down your food to most efficiently use all of that fuel. But if there isn't really a whole lot to process the fuel that you need, I would imagine that the organism would have to adapt to get in the fuel that it needs in any way that it could. And if there's no real barrier there, who's to say, the floodgates aren't open, and then you just have an excess of, whatever fuel there, and then it just gets stored into more mass and, substance in the animal. So, I mean, that would make sense to me. I'd definitely be interested in seeing the paper. But on the antibiotic line, there actually was a paper done in humans recently where you don't actually really fully recover from a regimen of antibiotics until 6 weeks after you've taken the antibiotics. So your gut microflora is, compromised obviously to kill the bacterial infection, but all those good bugs and stuff don't necessarily get replenished until a month and a half after you're already cured and better.
That makes sense. So like after you take a course of antibiotics, you have to wait that 6 weeks in order for your entire microbiome to regenerate?
Essentially, yeah. And there's things you can do to help. It's still very new. Some doctors recommend that you start taking probiotics, eating yogurt and kombucha and so on after an antibiotic regimen, but I don't think that's like normal practice necessarily. However, there is data coming out that it says, hey, your gut flora is not what it was after you've taken these antibiotics.
I see. All right. That's actually all the questions that I had, but whatchamacallit? Do you want to tell us more about like the Lactobacillus reuteri and then your work? Did you want to?
Yeah. I'll go into it. So obviously we went to Purdue together, and I was in a lab where I genetically encoded fluorescent proteins into neuronal cells to like detect oxidative stress. And so when I first came in to Scioto, I started learning everything I could about Lactobacillus reuteri. I started absorbing all this information and looking at what I know, which is the structure of proteins. And so I started to look into the different proteins that are on the surface of the cell that are binding to particular things. So interestingly, the Lactobacillus reuteri has receptors on its surface to bind to sugar molecules as well as collagen. And so this, like that kind of lets you know that this lactobacillus just kind of sits in this matrix of things that it's produced. Because as I stated earlier, it's also taking these sugars, binding to them and then attaching them to a chain and just making a very long chain of sugars. But then it also, binds to fibrous material like collagen. And so these bacteria are, what I believe to actually really start to make up that, fibrous structure and scaffolding throughout the entirety of your gut and your intestines, and that mucousy membrane to kind of protect you from any pathogens that could get in from bad bacteria and so on. So I really honed in on those particular proteins and started looking at different things that we could start testing. Started testing beta-glucan, which is a probiotic that is made by the bug. Unfortunately, I had a bad experience with the company that was selling us the ELISA kits, and their antibody wasn't specific at all to the beta-glucan strain. I mean, I was seeing increases, but then once I tested sugar and glucose and some other things just on the ELISAs, I realized that I could be seeing a little bit of false positive. There definitely was an increase, but we couldn't actually, publish anything and say, hey, like we're for sure that there's this extra beta-glucan. So it was really interesting to just kind of dive into those actual like surface proteins of the bacteria and look at, what is it doing? Like, what is this one bacteria trying to do within this larger scheme of, 1.9 trillion other bacterias? So it's sitting there and doing its part. So how is it affecting the other bugs around it? And I just learned, so much about bacteria in general and how important they are, and how much they're really just ever present, like in everything, and they've evolved with us. And when I started the job, my bosses recommended a book to me called, it's called I Contain Multitudes. I think the author is Ed Young. I could be wrong on that. But the book is called I Contain Multitudes, and it's all on the subject of bacteria. And it goes all the way back to the 1600s from like the first discovery of a bacterium, and then just throughout the entirety of science of what we know today. And it's a very interesting read, which I highly recommend. But
I'll go check it out.
It'll be, it's just something that, you don't really think about. Everything's like microscopic, and you just walk about your day without ever thinking about a bacteria. But, they're in your gut, they're eating your food, they're, processing your food for you. And another interesting tidbit, I'll send you the paper when I find it in my computer. But recently, when I was still at the company, probably in 2018, maybe early 2019, a paper came out that said approximately 2/3 of all drugs, all pharmaceuticals that are on the market right now, are actually metabolized by your gut microbiome and then changed into a different molecule. So basically, they might not necessarily know the mechanism of action of the drug. They just know that it works. Because I know, as from my experience as an organic chemist and learning about the neuroscience side of everything in graduate school, you're really like trying to make a drug that's going to have the best binding affinity in the receptor in the brain. But before it gets to that point, it has to go through the stomach, go through the gut, and then get metabolized by the bacteria before it even gets into the bloodstream to cross the blood-brain barrier. So I just found that paper so fascinating to me, because everything that we think we know about pharmacology, could. So.
All right. The more you learn, the more you realize how little you know.
Exactly. Yeah, for sure. Yeah. I don't really have a whole lot else unless you got more questions, but.
I don't have any more questions, but. Yeah, so I guess we'll end the recording there. And at the end of every single recording, I end up saying this for all my videos. Don't forget to breathe, because I forget to breathe a lot. So don't forget to breathe.
Good advice.